Hello everyone, a very warm welcome to Assist Biology. In this video, we will discuss about genetic disorders and pedigree analysis. I am Ashish Kumar Das, a student of MSc Animal Biology, University of Hyderabad. This will be the fifth part from the chapter Principle of Inheritance and Variation. If you have not seen the first four parts, I will mention the link in the description box so that you can watch the video. If you are new to this channel, please subscribe the channel and press the bell icon for getting more updates. So before starting, we will see what we will learn today. We will see what is Mendelian and chromosomal disorders. We will see what are the different types, different examples of the Mendelian disorder. What are the patterns of inheritance of disorder, that means how they are transmitted. Then what are the different examples of chromosomal disorder. Then we will see what is pedigree analysis and how this is becoming a helpful tool to trace the inheritance patterns of the diseases and also the risks of having an affected child. That means if the parents are affected, then what are the chances that the child will be affected. We will see this and many more. So first of all, pedigree analysis. So as the name suggests, analysis means it must be analyzing something. So a analysis that shows the occurrence and appearance of a phenotype. That means we will see how the disease is appearing, that is what are the chances of the occurrence and also what are the risks of having an affected child of a particular gene or chromosome from its ancestor from one generation to the next, and most commonly in human. Okay, so how the defective allele or the character is transmitted from the parents to the offspring, and most commonly we will study this in human. Then one question arises, why this is necessary. This is necessary because in case of plant, we can't do the test cross, back cross, etc. So controlled crosses cannot be performed. So the inheritance pattern of traits in human cannot be studied by crosses. Okay, and that's why means the controlled crosses can't be performed, and the progeny is produced very small in number. That means usually in case of that is Drosophila, you can take bacteria, you can take one mating will give you number of childs, or you can say number of offspring, but here very small number of offspring, that is usually one is produced, and that also is taking very long time. So representation, how the things that is the parents, childs, etc. are represented in case of the pedigree analysis. So first of all, you can see male is the square, female you can see the circle, then the mating, a horizontal line between the male and female, parents and children. So you can see these are the parents, these are the children, that is one boy and one girl in order of birth. So a boy is the first, that is from left to right, in the order of birth from left to right they are represented. Okay, then you can see this is also another representation to represent the childs, that is the children. You can see dizygotic twin, that are the non-identical twins. You can see monozygotic twins. So in case of monozygotic twins, there is a horizontal line like this, but in case of the dizygotic twin there are no line. Okay, then you can see sex unspecified. So this is also another way, that is sex unspecified. You can see like a tilted square or like a rhombus, you can see. Then again the number of children of sex indicated. So number of number of children of sex indicated. So these are the number of children. So 2 boy and 3 girl. Then again you can see the affected individuals, they are the darkened square and circle. Heterozygote for autosomal recessive. So for autosomal recessive, the heterozygote is like half sudden and half empty, and for sex-linked recessive you can see there is a dot in the middle. Then you can see death, there is a line. You can see the abortion or stillbirth, and the sex unspecified. Then you can see propositus. So propositus means considering which you are constructing the family tree. Okay, so considering this individual, the family tree is constructed. Then you can see method of identifying person in a pedigree, that is from the prepositus is child to in the generation. So prepositus is child to, that means by considering the child to, you have to track the inheritance pattern. Then you can see coincidence mating, that is the, or coincidence marriage, you can see that is the marriage between the relatives. So here you can see a pedigree analysis, that is the first generation, this is the second generation, this is the third generation. You can see the affected female is there, affected males and affected female are there, again the affected female and 2 affected male are there. So on the basis of order of birth, they are represented from left towards the right. Okay, you can see example of sex-linked recessive. So sex-linked recessive means, or you can say X-linked recessive, the defective allele is present on X chromosome and the and the that means you require 2 allele to show the digit. So X link recessive, so we'll write this as X dex. So as this is recessive and X link, I will write this as X dex and X dex, and the male is normal here, so we will write X and Y. Then you can see this is X dex, okay, X dex, X dex. Then you can see here the male are unaffected, so they will be XY. Female is unaffected, they will be XX. But here the female is unaffected, but the male is again affected. That's why this will be X dex, because the female will inherit one of the allele from the mother. Then you can see male is affected, so male is affected means X dex and Y. Then female is unaffected here, so it will be X dex X, or it also can be X dex and X, sorry, it can be also XX or X dex X dex. The male and female, you can see they are affected, so male affected will be X dex and Y, X dex and Y, and female will be X dex and X dex. So again you will get, you can see also X linked dominant. So X linked dominant means if they are having only one allele, the digit will be shown. So here also you can track the inheritance pattern. Now come to genetic disorder. So how we will do this? The genetic disorder, these can be grouped into two categories, that is the Mendelian disorders and the chromosomal disorders. So first of all we will see about Mendelian disorder. So Mendelian disorder, due to alteration or mutation in a single gene, and it can be dominant or recessive. That means dominant recessive we have already studied in Mendelism. That means dominant means if any one allele is defective, then the individual will show the disease. And in case of recessive, you need 2 copies of the defective allele. They are transmitted from one generation to another by following the Mendel's principle of inheritance. That's why they are called as Mendelian disorder. That means they will follow the Mendelian pattern of inheritance. Then the disorder can be autosomal, for example like cystic fibrosis, sickle cell anemia or phenylketonuria. So why they are called as autosomal? Because the defective allele are present on the autosomes. Then sex linked, as in hemophilia, color blindness or myotonic dystrophy. Why they are called as sex linked? Because the defective allele are present on the sex chromosome. Now you can see here the autosomal dominant and autosomal recessive patterns. So here you can see the affected parent, unaffected parent, and how they are transmitting the characters. So autosomal dominant means here you can see, so this is affected parent, so autosomal dominant, let this be capital A, small a, and this is unaffected, so this will be small a, small a. So all the affected individual will be having capital A and small a, these two, and the unaffected individual will be small a, small a. Here also autosomal recessive, so capital A and small a will be the carrier parent. Here also this will be carrier parent, capital A, small a, and you can also see the unaffected child here. That means it must be having capital A, capital A. So here the small is, that is the recessive allele. So why I have considered here the capital A is the defective allele, because this is dominant, and here this is recessive. That's why we are considering the small a is the recessive. Then you can see the affected child, that means having small a and small a. Then come to hemophilia. So hemophilia is a sex-linked recessive disorders. That means the defective allele are present on the X chromosome. So defective allele are the X chromosome, are on the present on the X chromosome. The defective allele produces a defective protein. That means the defective protein means this is a part of the cascade of proteins involved in blood clotting. As you have learned, blood clotting is a long process involving 12 factors. You no need to remember the names of the factor, but this is a very long process, and this is called a cascade process. That means step by step. So if one of the protein is defective in that process, so the blood clotting will be affected, and due to this reason the clotting of blood abnormally delayed, so that even simple small clot will result in excessive bleeding. So here you can see the more males than females are suffer from this disease. Why they are this like this? So what happens? If the male are affected, so they will be XH and Y, and if female are affected, they will be XH and XH. So female need 2 copies of the defective allele. Female need 2 copies of the defective allele, and 1 copy will come from the affected or carrier female, or 1 copy will come from, another copy will come from the affected parents, that is affected father. So the affected father are unviable, that means they will not be able to reproduce in the later stage of the life. That's why chances of the female will be to be hemophilic is very less. Then the homozygous female is carrier. So sorry, this will be heterozygous. Okay, heterozygous female will be carrier. That means the XH and the X. This is the heterozygous female, and they pass on the disease to some other, that is some of her sons. Then the queen Victoria is the, that is the carrier of the disease. So this is a historical aspect. So here you can see in this diagram, or you can say pedigree, the carrier female is there, the normal male is there. So carrier female is always affecting his son. So the son or the carrier female will be affected. Okay. Then the normal male is also there. Some of the son of the carrier female will be affected. If the female is carrier, that means XH and X, some of the son will be affected. However, if the female is homophilic, that means XH and XH, all the son will be affected. So here the female is carrier, half of the son is affected. Then you can see the normal female here, and normal female means X and X. So this is causing a carrier female and the normal male. So here the male cannot be carrier. As you can see, the male cannot be carrier here. If that means male are having only one X chromosome, and if it is affected, that is if it is defective, the male will be affected. So you can see here this. So you can see the inheritance pattern of the X-linked recessive allele. So father affected, mother unaffected. So the affected father will transmit the disease to the, or make the daughter carrier, and the son will be unaffected, because the son will always inherit the Y allele from the father, Y chromosome from the father. So there is no chances of the occurrence of the X recessive trait. So the carrier will be the daughter, or the daughter will be the carrier. But if the mother is the carrier, the son will be affected, okay, and the daughter will be the carrier. So this is the inheritance pattern. Male cannot be carrier, because what happens, the male are having only one X chromosome, and if the defective allele is there, they will be, that means affected. Then come to cystic fibrosis. So this is caused by recessive mutant allele on autosome, that is the chromosome number 7. So it produces a unique glycoprotein, that means the carbohydrate moiety is attached to protein, that leads to formation of mucus. Formation of mucus in the, that is the abnormally high viscosity mucus in lungs, in our various body parts. So this type of mucus interfere with the functioning of many exocrine glands like sweat gland, liver, pancreas, etc. and lungs also. So if mucus deposition will be there, so the normal transport will be affected, because the mucus will block the pathway. So the mucus accumulates in the tubules of lungs and makes them susceptible to infections leading to bronchitis. So here you can see the healthy airway is there. This is the cystic fibrosis airway. So the healthy airway, air can easily pass, but the cystic fibrosis, this mucus accumulation, this is the mucus. So this mucus accumulation blocks the pathway. So the liver and the pancreas show impaired function due to the mucus. That means the liver will produce less bile, and the pancreas will produce less digestive enzymes. Then come to sickle cell anemia. So this is caused by recessive mutant allele on chromosome number 11. So this is also a autosomal disorder. Then the mutant allele causes the substitution of the glutamic acid by the valine, so at the 6th position of the beta globin chain. So the normal human being is having that is the two chains, alpha chain and beta chain, 2 alpha chain, 2 beta chain in the hemoglobin, and if they will be affected, that means the 6th position of the beta globin chain is affected. So this will show sickle cell anemia. So normal blood cell is the biconcave shape, but the sickle shape is in case of sickle cell anemia. The defective hemoglobin undergoes polymerization under low oxygen. So you can see that these are the defective one. So they will undergo polymerization, and they will clog the pathway under low oxygen tension, and changes the shape of the RBC from the biconcave cells to the sickle cell elongated cells. So the disease is controlled by single pair of allele, that is HbA and HbS. So HbA is normal, HbS is affected. So of the 3 possible genotype, that is the 3 possible case can be HbA, HbA, can be HbS, HbS, and it can be HbS, HbA. So only one, that is HbS show the digit. So you can see the normal hemoglobin HbA gene, that is the CTC GAG is present in the DNA, and here CAC GTG, these are the sequence of the nucleotides. We will study this in details. It's present. Then you can see the mRNA, that is GAG and GUG is the affected one. So glutamic acid is there in the normal HbA peptide, and the in HbS peptide, that is sickle cell peptide, you can see the valine is present. Then come to phenylketonuria, that is PKU. So this is caused by recessive mutant allele on chromosome number 12. That means this is also autosomal recessive disorder. So the affected individual lacks an enzyme, that is the enzyme name is phenylalanine hydroxylase. So the phenylalanine hydroxylase enzyme is lacking in case of the affected individual, and catalyze the conversion of phenylalanine to tyrosine. So the phenylalanine hydroxylase is responsible for catalyzing the conversion of phenylalanine to tyrosine. So both of these are two amino acids. So phenylalanine is metabolized into phenyl pyruvate and other derivatives. So if the phenylalanine hydroxylase is not there, so this is catalyzing the that is the conversion of phenylalanine to tyrosine, but it is absent. The phenylalanine will be metabolized into phenyl pyruvate and other derivatives. So you can see the symptoms of the PKU. So accumulation of these chemicals in the brain will lead to mental retardation, and these are also excreted in urine because the kidney is not able to absorb this. So the deformed face, you can say, the teeth, mouth, and the foot, hand, these are the symptoms. Deformed shape, you can see the swollen foot, etc. Now come to chromosomal disorders. So caused due to absence, excess or abnormal arrangement, that is structure of one or more chromosome. So if chromosome is present in more in number, or they are less in number, or they are having abnormal arrangement, so they will cause the chromosomal disorder. So such abnormalities lead to serious consequences. So Down syndrome, this is caused when presence of extra copy of chromosome number 21, that means the trisomy of the 21, and this was first discovered by Langdon Down. What are the symptoms? Short stature, so the body will be shorter in height, short stature, small stature, small rounded head with a flat back, the back will be flat shape, partially opened mouth and furrowed tongue, the tongue will be furrowed, that means it is having like furrows, like you can say a little bit of like a swelling type. Then you can see the broad flat face with slanting eyes, the face will be broad and flat, eyes will be slanting in position, broad palm will be broad as compared to normal size, and the characteristic palmar crease, the palmar crease will be there. So the crease will be a little bit, that is of all of us having the crease in our palm, but here this will be abnormal. So here you can see the palm crease is the abnormal, many loops on the fingertips, then flat back, big and wrinkled tongue, that is furrowed tongue, furrowed means the perfect word is wrinkled, broad flat face, and the congenital heart diseases will also be there. So congenital means the diseases present from birth. So small and arched palate, epicanthic eye fold will be abnormal, like Mongolian people you can say. Congenital heart disease, mental retardation, that means the normal IQ will be less here. Then come to Klinefelter syndrome. So this is caused by presence of extra X chromosome in the male. So the normal male are 44 plus XY, but here one extra X chromosome is there, so it will cause 44 plus XXY. So although the individual is male, it bears a number of feminized character. So individual is male, but because it is having Y chromosome, but there are a number of feminized character. That means tall stature with feminized physique, breast development will be there in case of male also. So this is called as gynecomastia. So breast development in the male due to Klinefelter syndrome called as gynecomastia. Female type pubic hair pattern, the pubic hair pattern in the genital region will be, that means similar to female. Poor beard growth will be abnormal, that means the secondary sexual character will be affected, and it will be also sterility, so the individual will be sterile. So here you can see a tall stature with feminized character, this is a Klinefelter syndrome, and this is Turner syndrome, short stature and underdeveloped feminine character. So this diagram is of Turner syndrome, we will learn this, and this is the Klinefelter syndrome. So in case of Turner syndrome, this is caused due to the absence of one X chromosome in female, so they are called as XO female, or you can say X0 female, and they are having 45 chromosome. Normal is 46, that means autosomes 44 plus X chromosome XX, but here one X chromosome will be absent. So The neck will be webbed. These are the symptom: webbed neck, that means the webbing like shape in the neck. Rudimentary ovaries will be there, so the non-functional ovaries. Poor development of breast, the breast development will be abnormal. So here also you can see the secondary sexual character will be affected. Then you can see the shield shaped thorax also. The thorax will be shield shaped, and they are also sterile. Sterility will be there. So this is the end of the video. Thank you for watching. I hope that you liked the video, you enjoyed the video, and what I taught you will be helpful to you and you could easily understand that. Thank you for watching.